Okay, hello everyone. It is a gift to be opening another Global IMMUNOTALK. If I'm not mistaken, this is the Global IMMUNOTALK number 21 already. Yes, so I'm here today with the Global IMMUNOTALK co-organizer, Dr. Carla Rodlin from Yale University, and our great speaker today, Dr. Dan Littman from NYU. So as always, before introducing Dan, I would like to remind you about the goals of the Global IMMUNOTALKs, and remind you as well that these are not Carla's goals or my goals. We want these goals to be owned by everyone. And so we want this seminar series to benefit and inspire immunologists across the world in an egalitarian manner. For that, the talks are free, recorded in Twitter, and we do virtual, recorded in YouTube, and we do virtual discussions via Twitter. And as I was chatting with Dan recently, the reason why we do Twitter is because this allows asynchronous questions and answers. So you can ask questions right after the talks or even later for the people that watch the talks in YouTube. So, and we also like to increase opportunities for scientific learning without traveling. We will never get tired of thanking the speakers that make this possible. Not only the ones listed here, but also the ones listed in the previous flyer. We started these talks in April 22, so there are many speakers we have to thank that have shared their amazing science. And finally, I would like to remind you that our next speaker on September 23rd is Emily Truemel, who will be talking about intracellular pathogen responses in C. elegans. So make a note in your calendar so you can join us next Wednesday as well. So with that, I let Carla introduce Dan.
Thank you so much, Elena, and welcome everybody to a new Global IMMUNOTALK. It is a true pleasure for me to introduce today our Global Immuno speaker, Dan Littman. So Dan is the Helen and Martin Kimmel Professor of Molecular Immunology at the Skirball Institute at NYU, and he's also an investigator of the Howard Hughes Medical Institute. Now, Dan was born far away. He was born in Romania and came to the United States for his undergraduate studies at Princeton, and then moved to WashU, where he did his MD, PhD. And after that, he went to New York City and joined Richard Axel's lab at Columbia for his postdoctoral training. And it was when he was a postdoc that Dan first cloned what was known at the time as the T-cell surface glycoprotein 8. Now we all know this as the CD8, and then went also on to clone CD4. I really cannot imagine a better start for his independent career after having cloned these key, genes. And this, I'm sure in part allowed him to start his independent career at UCSF, where he raced through the ranks. His lab continued to do fundamental discoveries, such as, for instance, identifying that CD4 and CCR5 function as co-receptors for HIV. And we can continue to name some others, such as, for instance, the identification of RR gamma T as a key transcription factor in the differentiation of Th17 cells, as well as the identification of specific microbes, such as SFBs, as inducers of Th17 cells in the gut. I think these fundamental discoveries really took Dan to the gut, and today he's going to delight us, I'm sure, with recent findings on homeostatic and pathogenic cell interactions at the gut microbiota-mucosa interface. As you can imagine, based on his fundamental contributions to the advancement of science, Dan has received multiple awards and has been elected as a member of both the National Academy of Science and the Institute of Medicine, now known as the National Academy of Medicine of the United States. Now, Dan, on a more personal note, I was actually fortunate to meet you when I was a postdoc with a very good friend of you, Greg Lemke. I'm not sure if you remember, but I will never forget you had come to San Diego to give a talk at the Scripps, and I was able to get some time in your busy agenda to discuss my findings. I was entirely new to immunology at the time and was truly inspired by our conversations. So thanks a lot for accepting this invitation.
Well, thank you, Carla. It's really wonderful, and it's fantastic what you and Elena are doing. It's basically bringing immunology to the entire world, and it's something unique. It's one of the few good things that have really come out of this pandemic. So thanks again for including me in this. It's wonderful.
Thank you so much. So now, before you start, then, we love to learn a little bit more about our wonderful speakers. And you've been a scientist for a long time. You have had the chance to see many, I'm sure, fundamental discoveries. So before you start, could you share with us what have been the most impactful scientific discoveries, either in general or in immunology, that impacted your career?
Well, there are different aspects of discovery that impacted me personally. Certainly, from the side of immunology, the whole concept of tolerance and of antigenic specificity, where the things that really drove me towards immunology, things that I learned about when I was in college. But what actually really shaped my own future was the discovery of mammalian cell transfections. And when Wigler and Axel first published that it's possible to transform mammalian cells with defined pieces of DNA, as they did with herpes virus TK back in the late 70s, that's what really convinced me that I needed to work in that area. And I was very lucky to be able to get a postdoctoral fellowship in Richard Axel's lab, where I met your mentor, Greg Lemke.
Yeah.
So
wonderful.
That really transformed a lot of studies, including studies in immunology, basically allowing immunology to now be much more accessible through mammalian genetics approaches.
Exactly. No, that's great. Thank you. Thank you so much for sharing that. We are all really looking forward to your talk. So if you wish, you can now start sharing your screen, and we are all ears.
Okay, thank you so much. All righty. Hopefully this will be the right share. Oh. Did this come up? For some reason, I got multiple screens on here. Did it come up? No.
This is perfect then. It's
it is.
Perfect.
Okay.
Yes.
Okay. Fantastic. Again, thank you so much. So what I want to share with you today are a couple of stories related to the homeostasis at the mucosa, in the intestinal mucosa. This really lovely picture taken from clarified tissue by Jimmy Talbot in my lab is just an illustration of how rich the gut mucosa, in this case the small intestine, is in cells of both the nervous system and the immune system, in this case with GFP and macrophages and dendritic cells, and staining for neurons. And I'll come back to that in the second part of my talk. So we know that in order to establish homeostasis, there needs to be interaction between multiple cell types within this very complex ecosystem in the gut. And I shouldn't call it one ecosystem. There are probably many different ecosystems depending on where in the gut you're actually looking. But it's going to involve, of course, the microbiota, the complex microbiota that's in the lumen that can interact in certain cases intimately with the surface of the intestinal epithelium. You have epithelial cells, myeloid cells, innate lymphoid cells that I'll come back to and I'll talk about that also form cryptopatches. There are many different stromal cell types, different types of fibroblasts, and then there are the neurons and glia in the gut as well, of course, as endothelium of both lymphatic endothelium and vascular endothelium. So we need to really have an understanding of all of these components to be able to eventually understand how everything works in concert to maintain homeostasis, and to understand how breakdown of any one of these can contribute to certain diseases. So for the first part of my talk, I'm going to focus on microbiota-induced T lymphocytes and how they contribute to homeostasis. And the second part of my talk, I will discuss a particular subset of neurons in the gut that interact with type 3 innate lymphoid cells to maintain homeostasis. And these stories are studies done by these people, oops, excuse me, shown in this figure here. So the first part will be, when I move my mouse, which I shouldn't do, I'll use this. So the first part, parts that are together are about the induction of regulatory T cells and pathogenic Th17 cells in response to certain microbes, work that was started by Mo Xu, who's now in Beijing at NIBS, and Maria Pokrovsky when she was a graduate student, and now continued by Ranit Kedmi in our lab. And then there's work of Junyoung Lee on serum amyloid A proteins involved in Th17 cell differentiation. And I'll just briefly mention a cancer story that he's working on. And finally, I'll tell you about the work of Jimmy Talbot on these VIP enteric neurons in the regulation of the intestinal barrier. So starting out, this is what really brought us to study the intestine, was the discovery of the connection between the transcription factor ROR gamma T and the differentiation of Th17 cells. And of course, now we know that for Th17 cells to form, there's a requirement for a program that's directed by this transcription factor, which is a nuclear receptor. And Th17 cells can make a series of cytokines as shown here. Typically, we think of the IL-17A and IL-17F as being produced by what we might call homeostatic Th17 cells, but then other cytokines can also be made, in particular by cells that can have pathogenic properties. In particular, interferon gamma can be made by a very important subset of Th17 cells. And these cells typically are most enriched at mucosal surfaces, where they protect us from a variety of different potentially pathogenic microbes, several of which are listed here, both bacteria and fungi. But at the same time, they also regulate commensal bacteria to maintain a balance of appropriate commensals in different parts of our gut. But at the same time, the Th17 cells are the cells that have been most commonly implicated as being the pathogenic functions in autoimmune diseases and in chronic inflammatory diseases. We know from psoriasis that IL-17 producing cells, both IL-17 producing T cells, gamma delta T cells, and alpha beta T cells contribute to psoriasis, and anti-IL-17A antibody is very effective in the treatment of psoriasis. But we also know that Th17 cells are important in IBD, in various kinds of arthritis, maybe not so much rheumatoid, but ankylosing spondylitis and other inflammatory arthritides. And they might also contribute to neuroinflammatory diseases, but that is still not as well established. And there's also evidence, at least from animal models, that IL-17 producing cells, particularly Th17 cells, can contribute to the growth of, to the progression of colorectal cancer. So there's both a good side and a bad side to these cells. And that is illustrated through what we found when, quite serendipitously, Ivo Ivanov discovered a bacterium that induces Th17 cells. And what Ivo had noted was that when we purchased black 6 mice from Jackson Laboratory, they had very few Th17 cells in the gut. But when we purchased them from Taconic, they had a lot of Th17 cells. And then we were very lucky to start a collaboration with Kenya Honda in Japan. And Kenya was maintaining a colony of mice that were mono-associated with this bacterium, segmented filamentous bacteria. which are gram-positive spore-forming anaerobes. These bacteria had been described years before as forming these stringy filaments, particularly on the surface of the villi in the ileum. And we were able to show from this collaboration that mice from a Jackson laboratory had essentially none of these bacteria in their gut, and those from Taconic had these abundantly, shown here in the scanning EM. In a transmission EM, you can see how this bacteria interacts with the epithelium in the ileum. You can see it embeds itself. It signals in some ways we don't understand, leading to a actin polymerization reaction. Ivo Ivanov, now in his own laboratory at Columbia, with Pam Bjorkman, showed very beautifully that there can be release of material, of antigenic material from the SFB into vesicles, some kind of a new compartment of vesicle that they described last year. And that probably also that contributes also to the recognition of these bacteria by the immune system. And you can see the formation of these segments here. So when the animal is colonized with SFB, it protects it from pathogenic microbes such as enteropathic E. coli and Citrobacter rodentium. But it can also sensitize the animals to autoimmune disease. Oh, by the way, this is just a demonstration in our experiment with Kenya Honda that germ-free mice have in their CD4 T cells very few, if any, IL-17 and IL-22 producing T cells. And within a week after colonization with only SFB, you can see massive induction of these cytokine producing Th17 cells. So the downside is that Th17 cells can also contribute to autoimmunity, as I mentioned, and this is an animal model developed in the Benoit and Mathis laboratory a number of years ago. And in collaboration with them, Joyce Wu in their laboratory, together with Ivo, were able to show that SFB alone can drive this genetic model of arthritis. The animals typically don't get any arthritis when they're treated with antibiotics or kept in germ-free colonies. So there is both this positive and negative side to the induction of Th17 cells, and we'd like to understand how it is that SFB can induce this kind of disease in the sensitized animal, and to get a better understanding how there can be a systemic response that involves Th17 cells. And in the process, we found that Th17 cells are induced by SFB as antigen-specific T cells specific for SFB antigen in the draining mesenteric lymph nodes, and I'll come back to that in a moment. So we did we've done a lot of work on SFB that I'll very briefly summarize, but more recently we've also worked on another bacterium that colonizes mice that is thought of as a pathobiont, meaning a bacterium that can be symbiotic, but can also induce pathology in certain settings. And this is Helicobacter hepaticus. Kenya Honda had also shown that a cluster of Clostridia can induce regulatory T cells, and it turns out that H. hepaticus likewise induces antigen-specific Tregs, but at the same time it can induce Th17 cells, and these are a very different kind of Th17 cell, very different from the ones induced by SFB, that without having any genetic predisposition, as in the KBN model of arthritis, will cause inflammation in certain settings. And in particular, when these Tregs are disturbed in any way, and I'll show several examples of these, what happens is that they can no longer inhibit the Th17 cells from their expansion, and consequently, there is chronic inflammation. And I'll give you a couple of examples how this can occur. Now, there's a lot of interest in the so-called pathogenic Th17 cells, which probably correspond to what Federica Salusto has named Th1 star cells in the peripheral blood in humans. And just here, I just list a few things that we know about these cells. We know that the interleukin-23 receptor is required on the T cell for these kinds of cells to differentiate in animal models. And these are cells that often express interferon gamma. Gita Stockinger, in a lovely paper with fate mapping, showed that cells that at one point expressed interleukin-17A can later express interferon gamma and be pathogenic, and that transition requires the interleukin-23 receptor. Jean-Laurent Casanova identified several families that had homozygous recessive mutations of ROR gamma T, and these individuals were highly susceptible to mycobacteria like BCG vaccination, and that's because they lacked these Th1 star cells that express the chemokine receptors of both Th1 and Th17 cells, that is CXCR3 for Th1 cells and CCR6 for Th17. So these kind of mixed phenotype cells seem to be those that are required for protection from the mycobacteria. They make interferon gamma, and what's interesting in these individuals was that they could still mount normal Th1 responses to viruses, but could not mount these kinds of Th1 star responses. So we wanted to try to understand the difference then between the more homeostatic Th17 cells and these kinds of pathogenic Th17 cells. And this is where Moshe and Maria teamed up to look at animals that were colonized with both SFB and with the Helicobacter hepaticus. Helicobacter, unlike SFB, inhabits the large intestine. And there's a lot of work that's been done over the years from Fiona Powry, also from Alan Scher, showing that animals that were defective in interleukin-10 were prone to H. hepaticus-induced colitis, and that was a Th17 type, Th1's type of colitis. So the model that we set up was to generate TCR transgenic mice with T-cell receptors specific for SFB antigen and another T-cell receptor transgenic specific for Helicobacter antigen. And we can introduce these such that they are distinguishable by their isotype, and we can then follow their fate in animals that are colonized with both of these bacteria. Now, the SFB-specific T cells in the small intestine, as I tell you, become Th17 cells. So you can see they're almost all ROR gamma T positive cells, these non-pathogenic Th17 cells. On the other hand, the Helicobacter-specific T cells in a wild-type mouse largely become FOXP3 positive cells that also express ROR gamma T. And these are cells that are characteristic of induced regulatory T cells in the intestine, particularly in the large intestine. Those that didn't adopt this fate became follicular helper cells. Now, what was interesting was what happened when we put these into IL-10 mutant mice. These double positive Tregs virtually disappear. And instead, we see the appearance of these ROR gamma T only expressing cells. And these are pathogenic Th17 cells, very different from the SFB induced T cells. Now, Maria had also been studying the transcription factor CMAF in the differentiation of T cells, and found that these cells were absent in animals lacking CMAF in T cells. And indeed, when we knock out CMAF specifically in T cells using a Cre driver with FOXP3 driving Cre, and then gate only on Helicobacter peptide cells, Helicobacter-specific T cells using tetramers, what you see is that these tetramer-positive cells become Tregs in a control animal. In those lacking CMAF in the regulatory T cells, you can see these cells are lost, and instead we get the accumulation of these Th17 cells, which are CMAF sufficient. It's not due to a conversion of these Treg, putative Treg cells. Rather, there's now an expansion of Th17 cells that have the CMAF. And these animals, as well as the IL-10 animals in the IL-10 deficient, T cells in the IL-10 deficient mice give rise now to a colitis You can see here that there's rectal prolapse, there is lymphadenopathy in these mutant mice, and there is now this spontaneous colitis as shown over here. These cells, as I mentioned, are very different. The Th17 cells are very different from the conventional ones, the homeostatic ones that are differentiated in response to SFB. They're really distinct. They're both dependent on ROR gamma T, obviously, but they have very different programs. And we'd like to try to understand these better. So what we can say then is that a naive CD4 T cell, in response to certain microbial stimuli like SFB, will become these non-pathogenic homeostatic Th17 cells, but confronted with other types of bacteria, pathobionts like H. hepaticus, they adopt this fate here of becoming Tregs, and at the same time can also become these pathogenic type of Th17 cells, but the Treg hold these cells in check in large part through interleukin 10, possibly through ephraregulin as well. And we don't yet know if there is a pathway of differentiation from non-pathogenic to the pathogenic cells, but we do know that IL-23 and IL-10 exist in a mutually antagonistic relationship. So one of the questions we're really interested in is whether this might be a mechanism for commensal-specific tolerance. Has Helicobacter, is Helicobacter an example of bacteria that adopt to the host by inducing regulatory T cells, but have the potential to also be pathogenic, and potentially in individuals with inflammatory bowel disease, we may be able to discover these kinds of bacteria. Okay, so the question then is, the next question is, how is it that these different kinds of commensal bacteria can give rise to these different kinds of T cells? And Ken Yohanda has also shown that there are bacteria that can induce interferon gamma producing CD8 cells. I'll tell you very briefly about work that we've done on SFB through a schematic, and then I'll tell you some rather new work that we've been doing that is not yet published on the induction of regulatory T cells and how that may relate to the induction of these pathogenic Th17 cells. A few years ago, Teruyuki Sano, when he was a postdoc in the lab, found that Th17 cells that are specific for SFB are largely localized to the ileum, but there are also cells that are specific for SFB also scattered throughout the body, but only in the ileum they make the Th17 cytokine. So he investigated this, and what he found was that upon colonization with SFB, there's a rather rapid induction in a myeloid subset, we think they're macrophage-like cells, of the production of IL-23. And that IL-23 then acts on the type 3 innate lymphoid cells to release IL-22, which acts on receptor on the epithelium, and that leads now to a new program of gene expression by the epithelial cells, and the most highly upregulated genes are the serum amyloid A proteins. And that occurs within the first 3 days or so. At the same time, these cells migrate to the draining lymph node, and there they induce naive T cells with specificity for SFB to upregulate ROR gamma T and become Th17 programmed cells. And these are the cells that now distribute very widely throughout the body and throughout the intestines. But it's really only here in the small intestine, the ileum, where the SAAs are present, that the SAAs contribute to inducing IL-17A and IL-17F. And I'll come back to tell you more about the SAAs because, as you'll see, they can also induce a pathogenic program of Th17 cell differentiation. So it seems to be context dependent as to how the SAAs are functioning, but we think that they have really critical roles in Th17 cell differentiation. And I'll also come back to tell you about how ILC3 are regulated by enteric neurons. So how is it now that the other types of cells, particularly the regulatory T cells, differentiate in the draining lymph node where we first see them appearing as Helicobacter-specific Tregs? We know that dendritic cells drain the tissues where the antigen is present, in this case the microbial antigen, and typically use the chemokine receptor CCR7, and they interact with the ligand for CCR7, CCL21, or CCL19, and then go to the T cell zone in the draining lymph node for priming. And Ranit Kedmi decided to look to see which are the cells that are critical for the induction of Helicobacter-specific T cells, and performed single cell analysis, single cell RNA-seq combined with antibody, barcoded antibody sequencing, so-called site-seq, to look in the draining mesenteric lymph node for the different cell types that might be present there. And as you might expect, there are migratory dendritic cells that can be classified into CDC1 and CDC2s, and they express CCR7, as shown here. She was able to show that these cells can prime the proliferation of naive Helicobacter-specific T cells in culture when they are taken from animals that have been colonized with Helicobacter. But then she had a very surprising result when she looked in animals that were deficient for CCR7 in their dendritic cells. She colonized these mice with Helicobacter and transferred CFSC-labeled Helicobacter-specific T cells into these animals. And at day 3, she looked in the draining, in the colon draining mesenteric lymph node to look at T cell proliferation and also polarization of these cells or differentiation into regulatory T cells. So when she looks in control mice, this is what you see. You see proliferation of these cells, dilution of the CFSC signal, and upregulation of FOXP3. So this is what we expect. The surprise was that when we looked in the CCR7 deficient animals, lacking CCR7 in the dendritic cells, but we saw the same also with CCR7 complete knockout mice. And that's also very surprising. We actually saw that there was very strong proliferation still. So there was some CCR7 independent priming of these T cells. And turned out that these cells express ROR gamma T. This was in the absence of any migratory CDC1 or CDC2s here in the draining mesenteric lymph node, although there's still plenty of monocytes and macrophage-derived cells. When she looked later, in this case at day 10 in the large intestine, after the cells migrate back to the lamina propria, what she saw, as expected in the control mice, where these FOXP3 ROR gamma T positive regulatory T cells, but in the mutant for CCR7, instead she sees this massive accumulation of these Th17 cells, and these are these inflammatory pathogenic type of Th17 cells. And indeed, what you see now, if we look now at 17 days after gavage, this is a control animal, and this is an animal that is lacking CCR7, and you now see monocytic infiltrate in the villi and the lamina propria here, and there's clearly an inflammatory condition ongoing here. Now, this story has come together with something that was really unexpected, and it relates to some papers published by Greg Sonnenberg a few years ago, because Greg and others noted that type 3 innate lymphoid cells need to express MHC class II in order to prevent the expansion of inflammatory T cells like Th17 cells. So their conclusion is that there was a ILC3 intrinsic expression of class II that was regulated similarly to thymic epithelium, and they felt that this induces cell death of the activated Th17-like cells that are commensal specific. And we looked at this and saw something very similar to what they observed, but we interpreted it differently, because what we saw was when we looked at the ROR gamma T Cre driven MHC class II mice, loss of MHC class II in ROR gamma T positive cells, we saw the same thing I showed you before. Rather than having these double positive Tregs, instead we see the expansion of Th17 cells. So there's a defect, and these also express Tbet as these inflammatory cells that can also eventually make interferon gamma. So we think that the expansion is not due to a deletion of the Th17 cells, rather it's due to the absence of the regulatory T cells. So what is going on? And I'm just going to show you the model here because this is something that is still very much ongoing work here. But we think that these induced Tregs normally keep the Th17 cells from expanding, possibly from being primed. We don't yet know exactly what is priming these Th17 cells, except we know it's a CCR7 independent type of antigen presenting cell. But what's really fascinating is that the regulatory T cell seems to require two different cells for its differentiation. One is a conventional, probably a type 2 dendritic cell that is priming the cells, but at the same time, there needs to be antigen recognition of a cell that expresses ROR gamma T. It may be a type 3 innate lymphoid cell. It may be another type of cell that hasn't been yet fully characterized. Sasha Rudensky's lab last year published a paper, or early this year, published a paper on dendritic cells that can express ROR gamma T. But we don't yet know exactly what it is. But what we do think is that there is a ménage à trois established here, and that may be required for producing the kinds of cytokines needed for a regulatory T cell to differentiate. Interleukin-2, for example, which Greg Sonnenberg also showed, is made by ROR gamma T positive cells, by ILC presumably, as well as TGF-beta. So this is a very active area of investigation in our lab, but we think that it may be a very novel way of programming differentiation of a T cell. Okay, so I'd like to now just very briefly come back to the serum amyloid A proteins that I mentioned earlier. These are proteins that have been known as acute phase reactants for many years, very much like C-reactive protein. They're elevated in inflammation. They're apolipoproteins associated with HDL and also with retinol. They're made in huge amounts in the liver following infection or during inflammatory diseases. They're also elevated in tumors, in tissues, in autoimmune disease, and they're highly conserved. A number of receptors have been described, like TLR2, the formal protein receptor, but none of these is relevant for what I'll just briefly mention here. And the SAAs in human are known to be expressed at high level in inflammatory loci, and in inflammatory bowel disease, in this case, ulcerative colitis, you can see this expression of SAAs in the inflamed regions, but not in adjacent uninflamed regions or in healthy controls. So we're very interested in knowing how they might contribute to inflammation. And Junyang Lee, over the last few years, showed very nicely that the SAAs can contribute to direct differentiation of Th17 cells. Typically, as Gita Stockinger first showed, and also Vijay Kutru, it's possible to differentiate Th17 cells with a combination of interleukin-6 and TGF-beta. But what we see here is that if we block TGF-beta with an antibody, we can substitute now with recombinant SA1, SA2, or SA3 from the mouse and get induction of both IL-17A and IL-17F. But what's really interesting here is that when we do RNA sequencing, we see that the SAA-induced Th17 cells have what we might call much more of a pathogenic-like profile, similar to what I was just describing in these Th17 cells in the gut in the absence of Tregs. So it differs profoundly from the TGF-beta induced Th17 cells. And in the absence of SAA, in the model of colitis in response to Helicobacter and blockade of interleukin-10, you can see that the triple knockout mice lacking all three SAAs are largely protected from disease. And we know that this is due to a substantial reduction in the pathogenic Th17 cells that make both IL-17 as well as interferon gamma. So we also know that Th17 cells can contribute tumors. Michael Karen's group has shown that Th17 cells are involved in the growth of colorectal cancer. But at the same time, IL-17 has been proposed to, or Th17 cells have been proposed to also have anti-tumor functions in certain models. So the question was, do SAAs contribute in different ways to different kinds of tumors? I don't have time to go into this in any detail. I can just tell you that in the growth of colorectal tumors, the loss of SAAs results in substantially reduced tumor growth. On the other hand, it looks like the SAAs are required in certain models of tumors that are responsive to checkpoint immunotherapy. So this is just one example here, a system set up by Robbie Upadhyay in our laboratory of a KRAS P53 induced tumor that also is lacking mismatch repair, so that it accumulates neoantigens. When these are implanted in the lung, the animals are now responsive to anti-PD-1 immunotherapy. But what happens is in the SAA triple knockout mice, this tumor continues to grow in the face of immunotherapy, and the animals go on and die. We don't know yet whether this is due to Th17 cells, but that is one area that we're currently investigating. But we think that the SAAs do have important functions here we think in the tumor microenvironment, most likely to modulate the immune response against the tumor. Okay, this is just a summary of what I've been telling you, that SAA production by local epithelial cells can potentiate a homeostatic Th17 cell differentiation program, as found with SFB-specific T cells, but its production in other tissues can promote pathogenic Th17 cell differentiation and can contribute to autoimmunity. And the SAAs can promote tumor growth or immune control of tumors, depending on the context. Okay, I'm going to switch gears here, but what I'm going to talk about for the last part here has to do with innate lymphoid cells, particularly type 3 innate lymphoid cells. And just to remind everybody that the innate lymphoid cells are a relatively recent discovery to supplement what we know about natural killer cells that are also innate lymphoid cells that we've known about for a very long time. And they resemble T cells, particularly CD4 T cells, in their programs of expressing transcription factors associated with different cytokine programs, such as ILC1 correspond to Th1 through production of expression of Tbet, ILC3 make ROR gamma T, like the Th17 cells, etc. They're also lymphoid tissue inducer cells that are very much like type 3 innate lymphoid cells, and they're often clustered together with type 3 innate lymphoid cells, although they are known to have a separate differentiation pathway that peels off from the pre-innate lymphoid cell progenitors. And these also resemble each other. The T cells resemble the ILCs in their production of the different cytokine profiles. What we noted a number of years ago when Jason Hall and Maria Pokrovsky did RNA sequencing analysis of sorted innate lymphoid cells from the intestine was that the CCR6 positive ILC3, which differ markedly from the more Th1-like or ILC1-like ILC3 that are CCR6 negative, express a large number of genes that are also present in the central nervous system. And a postdoctoral fellow in the lab, Jimmy Talbot, became very interested in this, in particular because he noted that these so-called cryptopatches, which are made up of the lymphoid tissue inducer cells in the intestine, this is particularly in the small intestine, these, by the way, can be marked with a GFP knock-in at the ROR gamma T locus, a mouse that Gerard de Berle made a couple of decades ago when he was in the laboratory. You can see that these, there are a lot of neurons now that infiltrate these structures. So I should point out these cryptopatches receive signals from the lamina propria I'm sorry, they receive signals from the lumen of the gut, mostly from microbiota in the gut, and that leads now to the attraction of B lymphocytes and the formation of isolated lymphoid follicles. So Jimmy became very interested in these. Particularly, he noted this rich innervation of the intestine with neurons that go into the villi, as well as the myenteric plexus over here. There's, of course, intrinsic neurons that signal directly within the gut, and extrinsic neurons that whose cell bodies are outside of the intestine that can provide signals back and forth from the central nervous system. And what Jimmy noted was when he just stained with beta-3 tubulin for neurons and now looked at these ROR gamma TGFP mice, there were these neurons that went into these cryptopatches and looked like they were actually making direct contact with some of these cells. And when he then tried to characterize the neurons, what he found was that adrenergic neurons or dopaminergic neurons completely skirted around these cryptopatches, as did substance P neurons, for example. But those neurons that express vasoactive intestinal peptide, VIP, were the only neurons that actually track into the cryptopatches, shown here in double staining with beta-3 tubulin and the ROR gamma T positive cells. So he noted that the VIP receptor 2 was most highly expressed on these cells that make up the cryptopatches, the CCR6 positive type of innate lymphoid cell. So he decided to study these cells in particular and used some genetic models that have been established in the neuroscience community that allows for chemical regulation of individual cell types. And these are known as drug mice, designer receptor exclusively activated by the designer drugs, in which either activating receptors, G protein coupled receptors, or inhibitory receptors that bind this compound that's a muscarinic-like molecule called clozapine N-oxide, this molecule can then be used in these animals to exclusively activate or inhibit those cells that express these GPCRs. There are mice that express Cre recombinase, regulated by the VIP gene. So he bred these mice to mice that had been produced to conditionally express either of these receptors from the Rosa locus only upon expression of the Cre recombinase. So in that way, he could give these mice clozapine and look for activation or inhibition of VIP neurons. And then he looked at cells from the cryptopatches, these ILC3, the CCR6 positive ILC3, for their function. What he saw was that when an inhibitory receptor was activated, there was an increase in the production of IL-22 by these ILC3. And conversely, when he activated the VIP receptors, so there was an activating GPCR, now there was a really marked reduction in the production of interleukin-22 by the innate lymphoid cells. We know that IL-22 is very important for controlling various gram-negative pathogens in the gut, and most of the work that's been done is around using Citrobacter rodentium. So Jimmy looked to see what happens if there is a disruption of the VIP pathway in animals that were colonized with C. rodentium. Would there now be a disruption also in the barrier? And he looked in both directions, both inhibiting and activating the VIP neurons. And remarkably, when he inhibited the VIP neurons with relatively high levels of Citrobacter given to these mice, you could see that there was protection. So looking in spleen and looking in liver, you can see several orders of magnitude fewer bacteria that were recovered from these organs in these animals that in which there was inhibition of VIP production. There was very little difference in the amount of Citrobacter in the feces, however. And on the other hand, the activation of the VIP neurons, which now leads to less IL-22 production, made these animals much more susceptible to Citrobacter. You can see weight loss, and this is a relatively small amount. A low amount of Citrobacter was given to these mice. There's increased susceptibility with weight loss, and the animals now dying off, and though it's accompanied by increased levels of bacteria in the spleen and in the liver. So what we think from this work then is that, and other work that's been done by us and other groups, is microbiota, for example, bacteria like SFB will induce IL-23 production by myeloid cells in the lamina propria. The IL-23 then acts on these lymphoid tissue inducer cells, or a subset of type 3 innate lymphoid cells, so that they produce IL-22. And IL-22 then acts on the epithelium to upregulate molecules like reg3 gamma and DSAAs, that as I already told you. And that can now lead to increased protection of the barrier. On the other hand, when the VIP neuron fires, that will now inhibit the IL-22 production, and it will render the epithelium, the barrier, more susceptible to invasion by microbes. So Jimmy's question next was, why would this happen? Why would VIP be doing this? And what are the signals that can activate or inhibit VIP production by these neurons? And from literature from about 30 years ago, or even 40 years ago, primarily with dogs, it had been shown that shortly after feeding animals, there was an increase in VIP production in the intestine and in the portal vein circulation. So Jimmy looked also in mice at the VIP level in the ileum, in whole ileum, during the day and during the night. And the animals, of course, mice feed mostly at night and feed much less during the day. And indeed, what he found was it was at night during feeding that the animals had higher levels of VIP. So he decided to do an experiment to ask whether the IL-22 production by the ILC3 might be regulated by feeding. So in this case, what he did was to restrict the feeding. So the feeding was done only during nighttime in this case. So there was no ad libitum availability of food during the day. And then when he looked at the IL-22 level in ILC3, he saw a reduction in ILC3 during the feeding cycle in the middle of the night cycle shown here. But then he reversed this so that the animals were fed only during daytime, and then that they had received no food at night. And you can see there's a reversal of this, so that now during the daytime, there is an inhibition of the IL-22 production. And during nighttime, there's an elevation of IL-22. And this can also be seen in animals that have a mutation, animals that Jimmy made, in which he selectively knocked out VIP receptor 2 using RY gamma T3. So this will lack VIPR2 in type 3 innate lymphoid cells. And you can now see that in these mutant mice now, during the feeding cycle, they no longer can suppress production of interleukin-22. And the same kind of thing can be demonstrated now by forced feeding during the day. And you can see if test meal is given during the day, there's now inhibition of IL-22 production by type 3 innate lymphoid cells. Now, a couple of years ago, Ron Germain's lab published a very nice paper showing that interleukin-22 downregulates the expression of a variety of lipid-binding proteins and lipid transporters in the intestine, including CD36, the fatty acid-binding proteins, etc. And it leads to a reduced amount of triglyceride in the circulation. In this case, just one of the experiments using forced expression of IL-22 in these mice. So we wanted to know whether there might be an effect of this axis, the VIP signaling on IL-22 producing cells. Indeed, there was an effect on the absorption of triglyceride. In this case, what we did was to give the animals intragastric tritiated triglyceride, trilin, and did it just during the dark cycle or during the light cycle. And what we see is that we can now observe that in the feeding cycle, in the normal feeding cycle, what you see is that the animals will take up a lot more triglyceride in the serum now, and that we basically reverse this. If we feed the animals during the day, we can now reverse this pattern. At nighttime now, the triglyceride levels are going to be much lower. And this can also be seen here if we now use an anti-IL-22 antibody during the restricted feeding time, during the time that animals are fasting. You can see now an elevation of the triglyceride level as might be expected if the IL-22 level is going to be reduced. So what we think then is that the VIP elevation upon feeding is required to downregulate IL-22 production, excuse me, by type 3 innate lymphoid cells, but that leads now to increased absorption of lipid, in this case triglyceride, also fatty acids, and in that way regulates the host metabolism. But there's one other feature that was really remarkable and that Jimmy picked up by doing scanning electron microscopy and then doing 16S analysis of microbiota in these mice. And that was that during the dark cycle, during the feeding cycle, there was a VIP-dependent increase in the growth of SFB on the villi of these mice in the small intestine here. But in the mice lacking VIP receptor, specifically on type 3 innate lymphoid cells, this growth of SFB was lost. You see some stubs here, but it's almost as if these filaments washed away due to presumably antimicrobial activity driven by increased production of interleukin-22. So we can additionally then say that IL-22 contributes to the feeding, contributes to the absorption of the triglycerides. The VIP inhibition of IL-22 leads to increased triglyceride production, increased nutrients in general possibly, but at the same time there's an increase in SFB due to the reduction in antimicrobial peptides. And the last thing I wanted to leave you with here is the question of whether the VIP modulation can be applied in any way to metabolic diseases such as non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. And we're in very early days in studying this, but Jimmy now is collaborating with Harry Nguyen, a gastroenterologist working in our laboratory, to look in these mice. And in preliminary experiments using the chemogenetic approach, what they're finding is that the animals that fed a high-fat diet, of course, develop a fatty liver. But if there is now a modulation of the enteric VIP neurons, it is possible to prevent this kind of fatty liver accumulation. So like I said, these are still preliminary studies, but we think that this particular axis may be amenable for regulating lipid transport and the fatty liver disease. We still don't know what are the precise signals that might be involved in activating the VIP neurons, whether it's through intrinsic pathways here, or whether there's extrinsic signaling from the central nervous system. And that is one of the central questions that Jimmy is addressing now. I will stop on that, and again, acknowledge those who did this work. And I talked to you about work done by these folks. In particular, Henry contributed quite a bit to the work on the gastrointestinal pathology. Lina Kroling on the bioinformatics. Tariq Najjar has also worked on some of the aspects with Ranit on the nature of the cells inducing regulatory T cells. And these are some of the former postdocs and students who have contributed to this work. And of course, Kenya Honda with Koji Atarashi has been a long-standing collaborator on this work. And Fred De Beer helped us with the SAA mutant mice. So I'd like to thank you very much. This has been really a terrific opportunity to tell you about some of our work. And I want to thank again, Elena and Carla for this, for the invitation.
Thank you so much, Dan. That was wonderful. Thanks for your efforts and for such an elegant approach to really try to understand the complex biology of lymphocytes at the mucosal sites, right? And their responses to microorganisms, innervation, nutrients, and so forth. Thank you. I think Elena is going to remind us how to ask questions.
Okay. Thank you very much for having me.
Dan, that was a phenomenal talk. Thank you for sharing this, and especially all your exciting published and also unpublished data. Very intriguing. I mean, really a lot to think about it. So thank you, appreciate it. So, okay, so to ask questions for Dan, we will direct the audience to search for the account Global IMMUNOTALKs, and then find the tweet that is already there that says ask questions for Dr. Dan Littman here, and then reply to that tweet with your question and mentioning the hashtag Global Immuno. And then Dan will use the account Global Immuno Speaker to answer the questions. So thank you also for the effort to answer the questions from the attendees.
Glad to do that. Yeah.
Thank you, Dan.
So,
okay. Thanks very much.
We'll be in touch.
Okay.
Bye, everyone. Thank you.
Bye-bye.
Bye-bye.